Welcome everyone to this week's work in progress meeting and welcome Andrea. Andrea is an associate agricultural scientist and she will be presenting the mosquito genomics for vector control. I know a couple people had questions about that earlier today. She's an associate agricultural scientist at the Connecticut Ag Station and she joined there in 2018. She has expertise in population genetics, molecular biology, evolutionary genetics, vector biology, and experimental evolution. She studies patterns of genetic diversity in disease vectors to understand their historical and modern distribution and the implications of genetic variation on disease transmission. Her work initially focused on the yellow fever mosquito and now has expanded to study the northern house mosquito, the tiger mosquito, and the eastern treehole mosquito. Andrea, thank you for joining us.
Thank you. Thank you very much for inviting me to participate today. As it was just mentioned, my lab uses population genetics to investigate the movement of mosquito vectors and their evolutionary history, and as well as identify the genetic basis of epidemiologically relevant traits. Today, I want to talk to you about two of the lines of research that we are currently involved and that are related to challenges that vector control faces. I'm gonna start with the first challenge, which is the introduction of new vectors to areas where they had not been before. And this work has been done on the Aedes aegypti mosquito for over a decade in collaboration with Dr. Jeffrey Powell at Yale University and with funding from NIH. This work is also not possible without the collaborations that we have all over the world, which send us the interesting mosquitoes to the lab. Since we have here people from different systems, just a brief introduction of Aedes aegypti. It is commonly known as the yellow fever mosquito and is the main vector of dengue, yellow fever, Zika, and chikungunya viruses. This vector puts at risk for the diseases 2/3 of the world population. And although it was historically considered a tropical and subtropical mosquito, it has considerably expanded its range in the recent decades due to its association with humans and therefore with human migrations and trade. This paired up with the fact that the global weather patterns are changing and facilitating its establishment in areas that before were not permissive for this mosquito. So we study Aedes aegypti using different sets of genetic markers distributed along the genome of the mosquito. We use microsatellites. We use a genotyping array that contains approximately 25,000 single nucleotide polymorphisms across the genome that can do genotyping of 95 mosquitoes at a time. And we have also started to do whole genome sequences. Over the last decade, we had collected mosquitoes from across the Aedes aegypti distribution, and you can see some of those collections in this map, which has yet to be updated for the last couple of years, but as of this distribution that I'm presenting here, we had over 11,000 mosquitoes genotyped with the different markers. And so when we started analyzing this data that obtained from around the world, the first thing that we noticed is that we could cluster the Aedes aegypti from the world in two main genetic clusters. We did this with Bayesian clustering analysis. And what I show you here in the bottom is what we call then a mixture plot, or also a structure plot. And the way to read it is that every color, in this case blue and red, represent an inferred genetic cluster. Every vertical bar, it's an individual with the high of the color bar representing the percentage of ancestry from each of the genetic clusters. And the populations are separated by the black lines. And so this analysis, as I said, inferred two main genetic clusters across the world, which split very well between the African mosquitoes here in blue and the rest of the world, which is represented mostly in red. Now, what I want to highlight here is that in Africa, which was thought to be the native range of the mosquito, although we now know that it actually originated in the Southwest Indian Ocean before moving to Africa, this mosquito, it's a forest mosquito that breeds in tree holes and rock holes. It's a generalist mosquito that fits in a variety of mammals. Whereas outside of Africa, Aedes aegypti had become so adapted to humans that breeds in artificial containers and prefers to feed from humans over any other species. And so our genetic clusters pretty much separate both of the forms or subspecies of Aedes aegypti. Now, if we do a hierarchical analysis of the similar kind, what we see is that this genetic structure continues as we narrow down the regions. So here in the top, we have only the Aedes aegypti, which is the domesticated form of the mosquito. And you can see that this time we had inferred three genetic clusters in red, blue, and yellow that correspond approximately with North America and the Caribbean, South America, and Asia groups. And so, and we can do this over and over again as you move down in this slide. Sorry, my pointer is not working, but if you can see that you see structure all the way down to even the state level, here represented in the right in Mexico, in the left in the United States. And so what I'm trying to show you here is that at the regional level and up to the state level, we have a characteristic genetic signature of these mosquitoes. And this genetic signature can act as a fingerprint to identify the origin of the individual. Moreover, we have shown that this genetic signature is stable over a period of time that we had looked at, which is up to 10 years. And so what we can do with this is that we can use these genetic signatures to create a reference panel for the species from all around the world. And we can then use this reference panel with genetic association, with genetic assignment tests to determine where a mosquito that has just been introduced originally came from. And so this slide is just kind of a cartoon where we have the regions A, B, and C with specific genetic signatures, and every signature will correspond to a place in the map in the right. And so if we find a mosquito, I don't know, in the Antarctica, which is not going to happen, but that has a green signature, then we could determine that it probably came from North America. And how accurate the tracking the origin of the introduction is will depend on how complete our reference panel is. So the more mosquitoes we genotype, the more locations we have represented, the better would be our ability to track these introductions. And whether it works or not, I want to say yes, it does. We have been very successful at tracking introductions of these mosquitoes around the world. Here are only a few examples that had been already published. When the mosquito appeared in California in 2014, the occurrence of Aedes aegypti in Washington, D.C. starting in 2011. Most recently, Nebraska and Utah also reported Aedes aegypti, and we have done several trackings of mosquito introductions to Europe as well. Using these panels, we had also been able to determine that some of these introductions are in fact not real introductions, but more escapes from the laboratory next door or failure of the containment facilities. And so instead of creating a big alarm, we can confirm where the mosquitoes come from before we release the information. And this is possible because we not only have in the panel, reference panel of mosquitoes from the wild, but we also had genotype the colonies from some of the locations that we work with. These are introductions that we are recently working on from the last couple of years. And we're still finalizing the touches, but as you can see, these introductions are not always just like a short range expansion, but can also be introductions like in the case of Cyprus that came from a different country or even possibly from a different continent. Now, these genetic panels, as I said, also include laboratory strains. Because laboratory strains originally came from the wild before they were colonized, most of them retain the genetic signatures of the populations that they originally came from. But in the laboratory, things happen. And here is a figure from a paper we published in 2019 using our SNP chip to genotype several laboratory strains from fellow labs. And what I want to highlight here, and again, I don't have my pointer, but we genotyped several colonies from different labs for the Rockefeller and the Liverpool strains, which are laboratory strains that are frequently used for studies of vector competence or to understand the specific physiology or behaviors of the Aedes aegypti mosquitoes. And here what I'm showing you is a principal component analysis that groups the mosquitoes according to like their genetic variation, right? What we can see here is that we have the Liverpool strain in the bigger panel in 3 different locations. We have in the bottom left corner, one strain of the Liverpool group. Then we have just following up in the diagonal, you can see another group with 2 different Liverpool strains over there. And then if you go to the blow-up panel, which is like yet the 3rd cluster going up in the diagonal, we have another Liverpool strain. So 3 groups containing this technically the same Liverpool strain. And what this is telling us is that in the laboratory, not only the colonies could be evolving due to bottlenecks or the different ways in which they are treated, adapting to the lab, but also that contamination is frequent. And so a colony that you may think that is the same as your colleague in the UK, and when you start having problems replicating the results, is likely because the colony is not what you think. And these genetic panels can help you to determine whether that is the case or not. Here we had put also some of the wild collections of Aegypti just for reference, and you can see where the big A is there in the center. We have a lot of the Aedes aegypti wild isolates, and the Orlando strain right like above that. That's a collection that has come originally from Florida and has been now colonized in multiple labs, seem to be still close to Florida and seem to have not diverged as much as the Liverpool and the Rockefeller strains that I just mentioned before. And so I think that having panels like this are, again, useful, especially if you're trying to replicate the results from a fellow lab or even results you had done in the past to ensure that your resources are still the same. And so from this part, I want to emphasize that we can have comprehensive genetic panels to track invasion sources of invasive species, such as Aedes aegypti. But it's also important to remember that like really tight communication between the scientists performing the genotyping and the work with the vector control and public health departments is key to improve the vector control outputs. Because first of all, I would not know about the introductions if the mosquito control departments wouldn't tell me about them or wouldn't send me the specimens. But then once I have a result, and I communicate it back, then they can take action immediately. And so this communication, again, is really important to make things happen. And finally, with these panels, we can validate our resources, our laboratory strains to ensure replicability in all our experiments. So with that, I want to move to the next section of my talk, which is more focused on another of the challenges that vector control faces, which is the development of insecticide resistance. Before I do that, if there's any questions in the previous section, I can stop here, or I can continue and have the questions at the end.
Does that structure indicate that there's reproductive barriers between these strains, or is this more a geographic pattern that you think?
If you're talking, I assume, about the structure that we see, the correlation between geographic and genetic structure in the wild collections, correct?
Yes.
Yeah. So, well, I think that more than reproductive barriers, it's more related to the way that the species is expanding, which we have shown in a couple of papers that is probably a founder effect. So we get a population, like a group of individuals moving forward, right, and like jumping through whatever the means of transportation they get there, and then this bottleneck creates also a shift in the genetic diversity, a population that will start building a new population, and so on. And so there is more like not a reproductive barrier, but probably not a complete connectivity between the populations. So it's more like the gene flow rather than a barrier that is biological that prevents them from interbreeding.
But do you see introgression? Do you see evidence of hybridization between them when they do co-occur, or at the boundary?
Yeah, so there are areas in the world where we see more admixture patterns. For example, in the area of Florida and in New Orleans, the structure is not as strong as we would see it further inland. And so this is probably because we're getting a lot of inflow from the Caribbean, a lot of inflow from Asia, which we do have those genetic signatures all mixed up in there. If you remember the structures seen before, sorry, I hate to like slide here, but there. You see the Caribbean over there at the top, how you have a mixture between the red, the blue, and the yellow. So it's not that clean, right? And so there are areas where we do have more mixing. In Florida, in the bottom, like all the way in the B panel, you can see there like a more mixed, like mixture of colors compared to the other structures. So there are definitely areas where that mixture between the populations happen more often than others. But in general, for most of them, we can see a clean genetic signature.
Hi, I have a quick question, and thanks so much for such an interesting presentation and discussion of this work. I'm curious, just on sort of a practical level, how many mosquitoes you need to look at in order to get good resolution on sort of the origin of mosquito introductions, and then also how much background information do you need for a particular area to tell if it is an introduction or not? Thanks.
Yeah, so about the number of mosquitoes that you need to genotype, ideally you want to have a good number to represent the population that is like recently introduced. So a good number would vary between the markers that you are using. Like either you're using microsatellites, we usually like to have at least 20 individuals. But if you are using the SNP chip, which has 25,000 SNPs, or even whole genome sequencing, then even 5 individuals could be enough. Now, that said, when the introductions happened, we don't usually get that many, right? So in the case, we got an introduction in France, in a shipyard, and they sent 1 mosquito. And it is tricky because the assignment tests are based on the distribution of allele frequencies of the source population, right? So like when we are genotyping source populations, we want to have at least 20 individuals if we're doing SNP chip, at least 40 individuals if we're doing microsatellites to have a good idea of the allele frequency distribution. So when we get a single individual in the introduction and we want to track it, track the origin, it could be misleading because you're not getting a frequency, you're getting a single number. And so you can still tell with certain probability where the mosquito could have come from, but definitely having more than one will ensure that the accuracy of the assignment is the best. And so, yeah, I mean, it is possible if you have a lot of markers. That's the point, right? Like if you just have one mosquito, the microsatellites are probably not sufficient. You would have to go for the SNP chip, or the more markers you have, the more accurate your assignment would be. Okay, so now I want to tell you about a more recent project that we started a couple of years ago, which is focused on insecticide resistance in the Culex pipiens mosquitoes. And this work has been done in collaboration with a postdoc in the lab, David Giesbrecht, which is the leader on the next-gen sequencing and has been funded by the Ag Station Louis Magnarelli postdoctoral program. My co-investigator, John Shepard, who leads the mosquito control surveillance program here at the station, and my colleague, Nora Sarman at Utah State University in partnership with Vectec, which is a company that uses imaging to identify mosquitoes. And so I don't think that for this grad, I need to emphasize that insecticides are very important, especially because they are fast-acting tools that can be used during a public health emergency. However, because they are used widespread and some of the classes of insecticides overlap in their uses at home for vector control and agriculture, landscaping, these, as a consequence, can lead to an increase in insecticide resistance. And insecticide resistance can arise through the appearance of new mutations in the population that transform an individual from susceptible to resistant. It can increase in the population through selections if the mutations are already there at low frequency, but once the insecticide selection is applied, that mutation would increase in frequency, and therefore the overall levels of resistance in the population would increase. It can also arise from changes in behavior of the vector in response to the insecticide. For example, in this image, the mosquito, instead of resting on the top of the leaf, might rest on the bottom of the leaf, therefore sheltering itself from the direct exposure. And can also appear in a population through gene flow when alleles that are resistant in a neighboring population are brought into a population that was previously susceptible. And using population genetics, we can investigate which ones of these mechanisms are involved, and it's not only going to be one, but it could be a combination of all the above. And so for this, our study system is the Culex pipiens complex, which includes several species, some of the ones that are present in North America, and I will talk about them in a second. But Culex pipiens, the species of the Culex pipiens complex are the vectors of West Nile virus in the United States. And the virus cycles between Culex mosquitoes, it's maintained between Culex mosquitoes and birds. However, it is transmitted to humans and horses by behavioral changes in their feeding preferences or through bridge vectors. Horses and humans are actually ant hosts, which means they cannot propagate the virus. They're not part of the spiral cycle. However, they do end up being affected and carrying the disease if they receive the virus. In North America, this Culex pipiens complex is represented by two species, Culex pipiens and Culex quinquefasciatus. In this map, you can see the Culex pipiens is a northern species. It prefers to feed in birds, and it's able to diapause to survive the winters. On the other hand, Culex quinquefasciatus is the southern species. and it has a more generalist feeding habit, feeding from both mammals and birds. This mosquito cannot overwinter, so it's a more warm mosquito, if you want to call it that way. The distribution of these two species overlap in the United States. Approximately has been described between the 33 and the 36-degree latitude north. And in this overlapping zone, hybrids can occur. Now, to make things even more exciting, there is within the species pipiens, we have a form called molestus, which inhabits below ground. So it's found in subways, it's found in underground structures, and these mosquitoes have evolved the ability of the preference for mammals, right? Unlike the pipiens above ground that prefers to feed in birds, these mosquitoes feed in mammals, can go through the first egg-laying cycle without acquiring a blood meal, and they do not undergo diapause. So a lot of things going on with this mosquito in the United States, and the goals of our project are to define the extent of the pipiens and quinquina fasciatus hybrid zone more clearly, because we have now reports that this zone of the 33 and 36 degrees doesn't seem to be the real limit for the hybrid. So we want to do a better characterization of that hybrid zone. We want to determine whether pyrethroid resistant pattern, how are the pyrethroid resistant patterns in the Northeast United States, and identify what are the genetic basis of this resistance in the same region. So these are only the first goals of this project, but we have more that we will continue doing over the years, but I want to present you some results from these ones today. And so we started by using collections from across the United States obtained through VecTech. These collections basically were grouped in the West Coast, the East Coast, the Central, and the mountain regions. And they were genotyped initially using standard PCR that was developed by Smith and Fonseca in 2004 for targeting the ACE2 gene. So this gene has been determined to be useful for distinguishing between pipiens, quinquina fasciatus, and hybrids. And so it's the most economic way of starting the survey. And so in Utah, our colleagues took the mosquitoes, genotyped them all, and mapped them on the map that you can see here. And the first thing that they found is that we do find the presence of hybrids here in orange, beyond what would be the 33 and 36 hybrid band. Now, this is consistent with the reports from Cotera et al. In 2009 that suggest that the hybrid zone has been broadening over the years. The other interesting pattern that they found was that the hybrid index, which is the percentage of hybrids in the population in these locations, increased faster, or like the slope of the increase was faster in the east as well as in the central populations relative to the west and the mountains. And that this probably have to do with the local environmental conditions of these areas, as well as the history of colonization of when the mosquitoes came into the country and then established the secondary contact. This work is now being prepared for publication, and it will be submitted in the next couple of weeks, so you can soon read more details on this work. But this is the first approach we took at our collections. It did support the fact that the hybrid zone is not stable and that we should look for hybrids elsewhere. And so we wanted to get a more detailed picture of how these collections were related to each other, and how the percentage of hybridization that we could find in their genomes. And so for this, we took a subset of individuals, approximately 165, and did perform Illumina sequencing for the entire genome at 50x coverage. So what you have here is a principal component analysis of the mosquitoes that we genotype. And this is based on approximately 500,000 SNPs. And so what I want to show you here is that we have three main groups of individuals in this plot. We have the principal component one separating the two species. In the right, we have Culex quinquefasciatus highlighted in red. In the left, we have Culex pipiens, with Culex pipiens in a blue circle and Culex pipiens molestus in the orange circle. And we do see a separation in the second principal component between pipiens and pipiens molestus. Moreover, we see extensive variation within the pipiens group, and we can also see that some of these populations, in this case, the pink, the gray, and the blue that are towards the middle, precisely move towards the middle, and include Virginia, Suffolk, Virginia. We have Maryland, and we have California, Lincoln. And these three populations are somehow more leaning towards the quinquefasciatus side of the plot, suggesting that they might be hybrids, which would be consistent with what we are finding with the ACE2 gene. The other thing I want to mention from this plot is that, as you can see, beyond the hybrids, we have very little structure within the cluster of pipiens. If anything, we have some structure towards the north, but in the what we would be like the tri-state area and like more like the central northeast, if you want to call it that way, it seems like the gene flow is pretty even. We don't see structuring happening there. Okay. And so now I will focus on the northeast and the experiments that we have done with it to tell you more about the insecticide resistance. Based on the PCA and what we know from the ACE2 data, we wanted to establish what was the percentage of the genome of these mosquitoes that came from quinquefasciatus and pipiens along the East Coast. And so we used this measure called the hybrid index again, which does precisely that, and we plotted it in the map. And not unexpectedly, right, the mosquito populations with the highest quinquefasciatus percentage were more in the south. And as you move north, we see this quinquefasciatus ancestry dropping little by little. Now, in order to determine the insecticide-resistant phenotype of these populations, we decided to use a topical assay. And we did it for both bifenthrin and sumitrin. Today, I'm going to show you the results for bifenthrin. The way that, well, first of all, we decided to do topical assay as opposed to the CDC bottle bioassay because our ultimate goal is to link the individual phenotype to its genome and a specific genome region. And so this way we can control that all individuals are receiving exactly the same dose of the insecticide, and we can monitor them directly. And so the topical assay, as I said, we knock down the mosquitoes with cold, then we apply a known dose of the insecticide in their thorax and monitor them for up to 24 hours, determine which ones survive and which ones don't. And we have now 4 populations that have been tested through topical assay. Unfortunately, it has been more complicated to get live mosquitoes going in sufficient numbers in the lab, but we are still working on that. However, as you can see here, the results of the mortality by bifenthrin, we see a trend in which the mosquitoes that are highly resistant are in the south, in the area of Virginia, and as we move north, the susceptibility increases with the mosquitoes from the further north being the most susceptible mosquitoes for the pyrethroids. And as you recall, this is a very similar pattern to what we see with the hybrid index as well. Now, we don't have, as I mentioned, a lot of collections done yet for the topical assay, but the trend that we observe in insecticide resistance for pyrethroids, it's recapitulates what was reported by Bertis et al. In 2021 using the CDC bottle bioassay as part of the Northeast Regional Center of Excellence surveillance program. And you can see here in the right that how in general, pyrethroid resistance is higher in the southern populations of Culex pipiens than in the north. So together, these two studies confirm that susceptibility increases as you move north. Now, we are trying to understand what is the genetic basis of insecticide resistance in the region. And we know that one of the most best described genes related to pyrethroid resistance is the voltage-gated sodium channel gene named PARA. And specifically, a mutation called the KDR mutation or knockdown resistant mutation that in our case is that it's in the residue L1024 of the protein. And so what we did is we took the mosquitoes that we had done whole genome sequencing for, and we looked for that particular mutation and plotted it, and determined their frequencies in the different populations. And here are the results of that analysis, where we can see that the Culex Queen Capesiatus populations in general tend to have a very high frequency of KDR alleles. And as we move north, this frequency drops until we go all the way to Maine. Now, there is an exception there in the middle, that is the Manhattan population that has like 100% KDR mutations. That is the mutation, the population of molesters, so the pipiens that is underground, that is 100% mammal feeder. So there is a logic to this, and we find this trend very interesting. Potentially, we could use KDR to track the pyrethroid resistance of these populations. And we are increasing our numbers by doing more targeting sequencing of the KDR gene in the rest of our mosquito collections, which right now we have about 1,000 individuals from the region. So in order to figure out what else is affecting the insecticide resistant profile in these mosquitoes beyond the KDR gene, we are now taking a more genomic-based approach, and we are comparing the resistant populations from Suffolk with susceptible populations in Maine, as well as other results that I won't show today, comparing them with the populations in the South. And so this is just the first peek on that analysis, which is still ongoing. But what I'm showing you here is basically what David did was to look at regions of maximal differentiation in the genome that between the populations of Maine that are susceptible and the Virginia mosquitoes that are resistant. And this was done in 500,000 base pair regions along the genome. The greenish dashed line that you see there, it indicates the 2 standard deviation above the average FST value. So FST, the genetic differentiation again. And so what you can see here is that not much differentiation happened between these two populations in the chromosome 1, but we do see peaks of differentiation in chromosome 2 and 3. We're still exploring those from chromosome 3, but I want to highlight the chromosome 2 peaks of differentiation because it turns out that at least 4 of the main peaks that appear here, highlighted in the different colors, correspond very well with genes that are involved in insecticide resistance, more specifically pyrethroid resistant. We have a very nice red peak there indicating the location of the KDR mutation or the paragene. And we are picking up the UGT P450 clusters and other UGT genes that are related to, they are basically the detoxification enzymes that will help with the insecticide resistance or be involved in insecticide resistance. And so I think this is very promising, but we still have a lot to do. We have to narrow down the regions, see which specific genes are located there and which of the copies of these genes might be involved. And we want to confirm these signals using transcriptomics. We already performed an experiment with susceptible and resistant populations in the presence and absence of the insecticide. And we're hoping to start analyzing that data soon to see how it corresponds to what we are starting to find at the genomic level. Also, from what we expect to ultimately get from this analysis is to identify those loci related to pyrethroid resistance so that we can incorporate them in a genotyping platform that could be performed at low cost and paired with species identification loci. And the idea is that in a handheld device, such as the one from Oxford Nanopore that I'm showing here, we would have genes that cover both purposes, species identification, insecticide resistant, and maybe even the nature of the blood meals that could be do all at once for a very cheap price. Right now, we are troubleshooting that, and we had successfully work with KDR as well as ACE2 and ITS2 amplicons simultaneously, getting very good results for an approximate cost of $2 per mosquito. And we hope that this could be broadly available to mosquito surveillance departments or any other surveillance program that is interested on doing it, so that they could get real-time information of their populations, and that can be incorporated in their vector control decisions in a timely manner. And so in conclusion, I want to highlight that we reported the presence of hybrids well beyond the 33-36 latitude north, and that we do see genetic isolation between the north and south populations of the complex, so the Quinque fasciatus and pipiens groups. However, where they overlap, hybridization is possible, and that there is high connectivity among the populations in the northeast. We also showed that the pyrethroid resistance increases, sorry, this is the opposite way. The resistance increases as you go south, and that the frequency of KDR mutation could be a good indication for this resistance. Now, what implications does this have? Well, we know that the mutations are there already, and that any selective pressure could easily drive these insecticide-resistant frequencies up, and they could move from one neighboring population to another. And well, why do we care about the movement of these mosquitoes? Well, and the hybridization? Because as I said, Quinque fasciatus and pipiens have different diapause abilities, host feeding patterns, and insecticide resistant patterns, as well as it has been reported to have different vector susceptibilities for the different viruses. So if the alleles from Quinque fasciatus are moving north, that could imply a shift in the ability of the northern mosquitoes to transmit the disease. And therefore, it would be important to characterize such mutation and such movements of such gene flow in order to be ready and respond accordingly. So I just want to end there, and this is a more detailed picture of all the people involved in the QLX research. And as I said, this is still ongoing. So we hope to share with you more information pretty soon. Happy to answer any questions.
Thank you so much. Yeah, if anyone has questions, feel free to raise your hand or drop them in the chat as usual.
Hey, Andrea, thanks so much. That was a really nice talk. I had two questions. The first one about the PCA and with the Egypti and the different laboratory strains. Has there been any thought to try into metabolomics to understand the kind of functional impacts of that differentiation, or is that too big an ask?
I have not, I don't have experience with metabolomics, and I haven't like really thought about that. I had focused mostly on genetic signatures, and we do vector competence, so
Mhm.
we do know that they differ in their
Mhm.
ability to transmit disease, even between recently colonized strains and strains
Yeah.
that have been in the lab, that changes over time,
Mhm.
but nothing related to metabolomics to now. So
Okay.
you're welcome
And
to take over.
yeah, that's like a massive project. I'm just curious, do you see for the colonies that have been colonized longer, do you see further, are they more further differentiated or is it a patchwork?
Definitely. So the Rockefeller and Liverpool strains had been colonized for over 100 years. And as you could see just from the
Mhm.
few labs that we surveyed, they are very different from each other. So I would say in that way, we don't have enough sample size to say this is a pattern, but definitely seems
Yeah.
like the older strains do differentiate more and have more chances to I don't think that evolution is as big as the contamination within the lab.
So inbreeding
Um,
from other,
yeah, I think that there's a lot of strain contamination because mosquitoes are flying around and they
mhm.
may through the mesh if your mesh is not like thin enough, like close enough, and plus things that could just lay on the water
Yeah.
containers and things like that. The Orlando strain is a more recently colonized strain, and it doesn't seem to have diverged as much, but that could be just an artifact of which labs we survey as well. Laboratory colonies that I had started have not changed very much within the first 20 generations.
Okay.
So it depends on each lab and
Yeah.
yeah, on the history, I think.
Oh, super interesting. Okay. And then the other question on the nanopore sequencing, you mentioned the potential for blood meal analysis.
Yes.
How broad a range of species for host species do you think you'll be able to target?
So we have worked with the Culex pipiens blood meals as well, the Culex pipiens complex blood meals, as well as the Culex melanura blood meals. So we specifically are looking at mammals and birds at this point.
Okay.
I have to say that in general, blood meal analysis is tricky.
Yeah, you probably have to get
Um,
a really freshly fed one with.
Yeah, so
Mhm.
like it varies, which like your success will be usually about 50% if you're lucky, and it depends on how long the mosquito took the blood meal
Mhm.
and how it was preserved and if your primers were conserved enough.
Yeah.
And
Okay.
so it has been challenging, which is why I didn't talk about it today. I mean, you can include the primers, but whether you will be successful or not, it
Okay.
remains to be seen.
And then would that be possible in the $2 a sample window, or would that be additional?
Yeah, so David has done some calculations, and really, like if you just do one applicant, it was like 80 cents. And if you
Mhm.
add up, it keeps like adding a little bit, but it's not that much more.
Okay.
So once you have your library prepped, and we do in-house barcoding, and so it turns to be relatively cheap.
Okay. So even a 50% hit rate at the same cost per sample would
Yes, it
really
would be
useful.
worth just like,
Yeah.
additional information.
Yeah.
Ideally, we want to have more genes related to insecticide resistance, which is why we're waiting
Mhm.
for the whole genome analysis. But for now, we know that we can have KDR.
Yeah. Well, that's incredibly useful. Good. Thank you so much.
Thank you.
Sure. I have a question. I'm Jeff. That was really interesting. Enjoyed the talk a lot, actually. Many questions, but the one I want to ask you, I think, is have you tested for hybrid fitness effects? I mean, it's conflated a little bit with the KDR transfer, right? So if a hybrid has a high probability of having the mutation, then they're going to be selected for. But otherwise, do you see evidence that there might be a loss of fitness in the hybrids or a gain in fitness, or do you have any idea?
So we haven't done the experiments ourselves, but I know that there is a paper done by Ciota and Laura Kramer where they did some hybrid crosses, like they artificial hybrids. They claimed that in general, all the hybrids had higher vector competence, but it was like, they didn't measure their fitness themselves. I think that there's a lot more to do, and that we cannot take that experiment as like the gold standard. Regarding the fitness, I think I can say from the work that we did with Nora and the ACE2 hybrid screen in her and that paper, she compared the hybrid distribution with the niche prediction, where you would expect quincke fasciatus and PPNs to be present with areas of overlap, and where you see the hybrids and where you're not seeing hybrids. And it seems like in colder areas, you see less hybrids that you would really expect to see from just the co-occurrence. So like the hybrids are not everywhere where they could be. And this might have to do with like advantages under certain environmental conditions as opposed to others. But we don't have a direct proof besides that.
Thanks. Interesting.
What we do know at this point is that it seems like that KDR haplotype seems to be coming definitely from the quincke fasciatus background. And we're still working on the haplotype studying the haplotype movement, but it points to that way. Okay, well, thank you, everybody. It was really nice. I'm happy to answer more questions if you have any, or if you would like to follow up in an email or in other conversations. It's great to be able to reach out a new group of people and a new audience. It has been a great opportunity.
Thank you for speaking and presenting this week. Thank you everybody for coming. We have one more work in progress meeting this spring and summer, and that's going to be on June 25th. We are skipping the next one, so about a month from now. So we will see everybody then. Thank you so much.
Thank you very much.